Hassan, thank you very much for talking to an audience which is interested in host-microbe interactions and to the research center, Origin and Function of Metaorganisms. You're born in Cairo, you got your education in Miami, in Jena, Germany. You did a postdoc in Emory University. You're now here at the WiKo in Berlin. You will go to a Smithsonian Institution. I met you at a Symbiosis meeting in the UK, and you presented at that meeting a fascinating observation you made a few years ago. So maybe the first question goes to you, and how did you find the research question you are interested in, the symbiosis, the beetles? How did you get to there?
Okay, well, thanks so much for having me here, Thomas. It's a real pleasure. How did I find leaf beetles, and why did I become interested in their symbionts? I became interested in that specific question, or that model. It's because I was interested in folivores, or leaf-feeding animals. And they're an interesting group from a microbiological standpoint because they have, for the most part, very conserved microbial communities. Whether you consider ruminants, whether you consider koalas, the giant panda, they have conserved communities that do very similar things. They break down plant matter, they ferment, they supplement various nutrients. But of course, one of the shortcomings of these systems is that they're quite complex. And so you're dealing with operational taxonomical units that are an upward of 800 to 1,000. And so assigning individual functions to specific taxa is a challenge. And so for my postdoc, I wanted to work with a folivorous animal that is compositionally a bit less complex and is experimentally more tractable. Mm-hmm. And that's where I shifted my attention to insects, more specifically beetles, to address that question.
Mm-hmm. So what was the key observation which you published with, as a first corresponding author in Cell? What, if you summarize the few key messages, what this paper, which is really a heavy and an interesting and a paper which goes into the future and possible directions, what are the key messages of that paper?
Sure. Well, most of the work that we did on that paper you're referencing was actually based on histological observations that were 80 years old. And so we found this really interesting paper, very old paper that turned 80 2 years ago, published by a German scientist, Hans-Jürgen Stammer.
Mm-hmm.
And
where did Stammer live?
He lived in Erlangen.
In Erlangen,
and
okay.
he was a student of Paul Buchner, and so it's,
Buchner, okay.
And he comes from a lineage of symbiosis researchers.
Right.
But most of his work was forgotten, and a combination of recency bias that we have towards papers and the language barrier. And so his work, which essentially detailed all these organs within leaf beetles that, if you look at today, you immediately recognize as symbiotic organs or bacteriomes. And so we picked up from where he left off in 1936, and applied all the tools that are available to us at the molecular level, at the genomic level, the transcriptomic level, to understand just how complex the relationship is between leaf beetles and their symbionts. Mm-hmm. And so what is the main finding that we got from this paper is that leaf beetles, specifically tortoise leaf beetles, they possess symbionts that do a very specific thing, which is pectin degradation. Mm-hmm. Pectin, of course, is a big component of the plant cell wall. Mm-hmm. And so the mutualistic factors that define the relationship between leaf beetles and their primary and the symbiont is quite simple, and the manipulation of the model is amenable.
Mm-hmm. Mm-hmm. So from that key observation, which clearly showed a very strict metabolic dependence on the symbiont and the beetle, here at WiKo we occasionally ask ourselves, what keeps you up at night? And so what drives you right now with in the back having this very nice observation?
Okay, well, I think that's one of the funner questions to ask here at the Wissenschaftskolleg. I find it funny because very little keeps me up at night. I'm a very sound sleeper. But there are questions that that engage me for a long period of time, and one of them is how widespread are these mutualisms? Is it just restricted to tortoise beetles, or is that a wider phenomenon in folivorous insects? And what exactly can we extract from these mutualisms to other folivorous models like the panda, like ruminants? And so that's one question that I'd like to have an answer for fairly soon. And just exactly how adaptive is the impact of that mutualism is. So what exactly does it mean for an insect to rely on the plant cell wall degrading capacity of its symbiont? Is it to upgrade the carbon metabolism of the host, or is it to maximize the nutritional value of the diet?
I see that. I see, of course, it's super interesting and exciting. I see also that you are at a turning point of your career. You have to make major decisions, and the obvious question, which you probably ask yourself quite often, is this leaf beetles and the pectinolysis and nitrogen fixation genes in these guys. Is that something which will carry you through an academic career? Is that something you may have leave at some point? I mean, and that goes also. Pectinolysis, of course, has always a translational aspect. I mean, probably a lot of people would like to use, if one finds the enzymes, and can we use that then in a translational aspect? So what do you think about that?
Certainly, that's a great question to keep in mind always, like as you develop grants and sort of plot new directions. I think one thing that is central to this system is why symbiosis is a strategy for an animal to exploit folivory or to exploit leaves as a nutritional resource. The reason this is a question is because other leaf beetles lack the symbionts. Not all the leaf beetles, but some other leaf beetles lack the symbionts, but they possess the mutualistic factor within their own genomes. So they've horizontally acquired pectin-degrading genes, or genes coding for pectin-degrading enzymes, into their genomes. And so now at the center of this is the question of why keep a symbiont when you could potentially have the gene? What exactly about symbiont acquisition that relaxed selection to lose these genes? And so it's a question of horizontal gene transfer versus symbiosis as a mechanism for integrating exogenous genetic material.
Mm-hmm. So imagine now, you have possibility to employ people, and you can now develop different areas of research. Where do you want to focus on, and what's the most intriguing and interesting questions, starting from a very intriguing single observation? Where do you want to be in 5 years with the help of Grant and other people? Where do you want to be in 5 years, and maybe even where do you want to be in 10 years?
Right. So where do I want to be in 5 years? I'd like to be sitting here, maybe seeing you at a meeting, and tell you with some level of authority how widespread this phenomenon is. Wow. Is it just is it an isolated biological incident, or is it in fact representative of adaptations that animals need to acquire to exploit leaves as a sole nutritional resource? Leaves are everywhere, and the evolution of folivory in animals happened independent times. And my question is, did these 5 independent radiations into leaf feeding behavior involve similar steps, and where do microbes fit in these steps? Mm-hmm. And so that involves a fairly broad characterization of host-microbe interactions of these systems. Mm-hmm. And then going back to this very simple model that we have between Cassida rubiginosa, the tortoise leaf beetle, and its symbiont, Stammera, and sort of assign some of the tractability that the system can offer for broader implications of other systems.
Mm-hmm. What were cornerstones of this success? If one of your junior fellows here at the College of Life Science asks you, so what is really important? What would you tell your classmates?
Well, so first of all, our field is extremely nurturing. When we go to symbiosis sessions, we're fortunate to be part of a great community. That includes also having great mentors. And so that's one thing that I feel fortunate to have had. But what do I tell to my colleagues is that don't shy away from non-model organisms. Model organisms are great for our understanding of biology, but they're not ideal for understanding every question in biology. So leaf beetles were a great system to address the question of what mediates folivory, or what roles do microbes play in mediating folivory in animals. And this was possible to address in 2019, 2018, 16, because of the advances in
technology,
in omics,
yeah,
right? And it's just a very simple team was able to establish the molecular toolkit and the genomic toolkit to really, dissociate a mutualism.
I think that's very brave. Of course, also I like it very much that you see that moving away from non-model organisms. But it also needs, of course, then the challenge which you then and the many difficulties which you will find on your way. I mean, we did the same thing with Hydra, where we certainly a non-model system. It took me 10 years to molecularize it more or less and make transgenics. So then it's one advice would be move away from non-models. That's I certainly agree. But then it needs also a type of personality which is then able to somehow solve the many non-model specific problems by being open to novel developments, technologies, talking to other people, employ other people, etc. One of the peculiar features here of the WiKo is the interdisciplinarity, and so you come from very well equipped and intellectually equipped labs on symbiosis research. Now here we are confronted with historians of science and with social biologists and with philosophers, and so is that of interest, or does it enrich your way of you think now, going back to your tiny little specific problem?
I mean, it's humbling because you're surrounded by people who are, leaders in their fields, and it's exciting because you get to also explain your research to them in ways that you otherwise wouldn't in your small departments or in your lab. So you have to sort of paint your questions with somewhat of a broader brush. And so that's wonderful, but also teaches you to be a better writer. You're exposed to different forms of writing. You're exposed to different forms of thinking. And so that's indispensable. And that's sort of one thing that it's hard to find outside of a place like the Wissenschaftskolleg-Fellowship. And so...
Yeah, I'm very glad that you made it here. Thank you very much.
You too, Thomas. It's nice to coincide with your time here as well.
Thank you very much. Thank you.